Hello, friends. Welcome to my YouTube channel, The Microbiology Digest. Today, our topic of discussion is a bit different. Means we will not be discussing about virus. We will not be discussing about bacteria. We will not be discussing about any other type of microbes. Rather, a different type of infectious agent which is responsible for causing a typical type of disease in man and animals. We'll be discussing about those diseases and we'll be discussing about the type of infectious agent which is totally different from our earlier discussed topics. So, welcome to our discussions on prions. I am Dr. D.P. Bora. So, prions or prions, they are responsible for causing a different type of disease condition, which are collectively called as transmissible spongiform encephalopathy, or in short, they are called as TZE, transmissible spongiform encephalopathy. Now, prions or prions, these agents, they are infectious agents. They do not have a nucleic acid genome. They are structurally composed of a protein and they do not have any nucleic acid. So by definition, prions are proteinaceous infectious particles which are devoid of nucleic acid. Prions means these are proteinaceous infectious particles devoid of nucleic acid. So if we go back to the history, how these infectious agents were discovered, so it was very recent, so to say, but the search for these agents started long back when in 17th century a disease called scrapie, it was described in sheep. Then in 1950s, a specific tribe of human called Kuru, they were suffering from a particular disease, the only restricting to that tribe only. So this disease that is called as Kuru disease, this was studied by William Hadlow. Then subsequently, the other scientists also they were simultaneously conducting research on Kuru or the specific disease restricting to the Kuru tribe, and at the same time, another scientist in the year 1966, Gottschalk, he identified that this Kuru type of disease they are recorded in some other animals, which was suspected to be due to the ingestion of infected brain tissue. Then 1981 scrapie was studied and the fibrils were detected in mouse brains. Then in the year 1982, finally, one scientist called Stanley Prusiner, he could successfully establish a link between this similar type of disease occurring in human as well as in animals. So Kuru in human, then BSC, another type of disease which is will come to this disease later. It is a disease in cattle. So he could establish the relationship or similarity between these diseases. And because of this great discovery, uh, he was awarded with Nobel Prize in Physiology and Medicine in the year 1997. And it was Stanley Prusiner who first coined the term prions or prions. Now, what is a prion? So, as we have already discussed, this is simply a protein, but they are normal cellular proteins. If we say what is a prion, so this is a normal cellular protein which is present in the brains. And these proteins are predominantly they involved in the synaptic junctions or synaptic functions. But this normal protein, it is not responsible for causing these diseases or they are not responsible for transmissible spongiform encephalopathy type of diseases. Then what is the prion? So prion or prion as we have already discussed, these are infectious particles without any nucleic acid. These prions are a modified form of normal cellular protein. So normal cellular protein, it is written as PRPC, and the abnormal form which is responsible for causing these diseases, this is written as PRPSC. SC means it is from the disease scrapie. So now invariably, although it was taken from scrapie, so all the infectious prions responsible for causing diseases are denoted by PRPSC. Okay, this is the bad conformation. So normally what happens when this normal protein, cellular protein is present in the brain cell, it is not responsible or it cannot cause any disease. But if somehow there is alteration or structural alteration of this protein to PRPSC, then it becomes infectious. Okay. So that is why the normal protein is denoted as good conformation or normal conformation, and the bad conformation PRPSC is denoted as bad conformation or infectious conformation. Now, what are the characteristics of prions or prions? As we have already discussed, they are made from normal cellular proteins which gets converted to an abnormal one. They are extremely resistant to heat, radiation, and disinfectants. They are responsible for causing spongiform changes in the brain tissue. They typically have a very long incubation period, and because of this, before earlier to discovery of prions, this type of diseases were thought to be caused by some type of viruses, and these diseases with long incubation periods were studied under a separate heading called slow viral diseases. These proteins, prion proteins, another characteristics is that they can reach very high titer in brain tissue. In the brain tissues, these proteins may reach very high titer. The abnormal form of the protein, which is responsible for causing disease, that is the PRPSC, it is resistant to any form of digestion. Means they are highly resistant to proteases. Prions cannot induce any immune response because they are normal cellular protein. Only the conformation of the protein gets altered. So that is why our immune system cannot recognize this protein as a foreign particle. So that is why there is no immunogenic response against prion proteins. No antibiotics can cause or no antibiotics can cure prion diseases. And finally, these prion proteins, they are not easily decomposed biologically. Means they are resistant to all forms of available detergents. They are resistant to heat, they are resistant to radiation, they are resistant to proteases. Means they cannot be decomposed or they cannot be destroyed. Now coming to the pathogenesis, how prions they can cause disease. So as we have already discussed, prions are normal cellular proteins. So this normal protein, structurally if we see, the structure of the normal prion, it is mostly composed of the alpha helix structure of the protein. But when it gets converted to the bad form or the PRPSC, here instead of alpha helix, the predominant conformation is the beta helix type of conformation. So what happens when a particular host it ingests abnormal form of prion protein or PRPSC by any means, through by infected meat or by any means, this ingestion of the abnormal form of PRPSC, it will act upon the normal prion which is present in the brain tissue. So what it will do, this abnormal conformation protein or the bad conformation protein or PRPSC, it will start acting upon the normal cellular protein or normal prion protein, that is the PRPC, and it will initiate post-translational modification or alteration of PRPC to PRPSC. Means due to ingestion, it will act upon the normal protein, and the normal protein, due to action of the abnormal protein, it gets converted to the abnormal one. These abnormal proteins or abnormal prions or proteins, PRPSC, these proteins, they get accumulated in the brain tissue, forming a specific structure which is called as SAF, or scrappy associated fibrils. Then these scrappy associated fibrils, they are resistant to the action of proteases, and this SAF is actually responsible for causing transmissible spongiform encephalopathies in man and animals. Now, what are the diseases caused by these prions? So if we can recall, we have already discussed that diseases caused by the prions or prion proteins, they are discussed under a different category. Collectively, these diseases are called as transmissible spongiform encephalopathies, or TSE. So both human and animals are infected, may be infected with prion proteins. So, and they are responsible for causing some diseases. In animals, for example, in cattle, it causes bovine spongiform encephalopathy, which is also otherwise called as mad cow disease. Then scrappy is the another one, which is in sheep and goats. Then transmissible mink encephalopathy. Then chronic wasting disease of the ear. So these are the diseases caused by prions in animals. So in human too, these prions can cause some diseases. For example, Kuru, already we have discussed. Then CJD, it is Creutzfeldt-Jakob disease. Then FFI, fetal familial insomnia. GSS, Grassmann-Straussler syndrome. So these are the diseases in human which are caused by prions. And these prions or the transmissible encephalopathies, they are always fatal. Okay, once a person or individual or animal gets infected, so there is always 100% fatality. And the gold standard method for diagnosis of this type of infections or infections caused by prion proteins, the gold standard test is the immunohistochemistry. So immunohistochemistry can give the indication about the diagnosis of the diseases both in man and animals. So this is all about the prions in nutshell I wanted to share with you. So I hope informations will be helpful. If you find these are helpful, please subscribe to my channel and give your comments. And with this, thank you. Thank you very much.